Welcome to the PubMed Daily Review for May 14th, 2026. Today we scanned 5,742 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5, Cognition, Behavior, and Health Outcomes. This trend investigates the intricate links between cognition, behavior, and health outcomes across diverse populations and biological levels. This cluster covers 1,381 papers. Representative work includes cortical motor activity modulates respiration and reduces apnea in neonates. Drift in individual behavioral phenotype as a strategy for unpredictable worlds, and no interaction between physical activity and body mass index on chronic pain. A cross-sectional study. Trend 2 of 5, Nanomaterials for Catalysis and Energy. This trend explores nanomaterials for catalysis, energy conversion, storage, and advanced functional devices. This cluster covers 781 papers. Representative work includes engineering of edge-enriched nitrogen-doped porous carbon as a high-performance metal-free catalyst for acetylene hydrochlorination, aqueous imzine-assisted charge transport for sliding Cu/NCDC, triboelectric nanogenerators, and arbitrarily large area graphene suspension with ultra-low standoff for varying capacitance applications. Trend 3 of 5. Genomics, microbiology, and biological mechanisms. This trend explores genomic insights, microbial interactions, and fundamental biological mechanisms governing diverse life processes. This cluster covers 1,480 papers. Representative work includes TCP21 negatively regulates photomorphogenesis by integrating PHYB and HY5, mediated signaling pathways in Arabidopsis, optimization of the in vitro model of cardiac fibrosis, and genomic and evolutionary insights into a novel Neisseria meningitidis genogroup W clonal complex 11 variant with dual resistance to penicillin and azithromycin in Spain. Trend 4 of 5, Cancer Mechanisms, Biomarkers, and Therapy. This trend unifies research on cancer's molecular mechanisms, identifying biomarkers, and developing advanced, targeted therapeutic interventions. This cluster covers 733 papers. Representative work includes germline genetic testing among patients with pancreatic adenocarcinoma, a pancreatic cancer action network patient survey, development of immunoelectron microscopy for VSN detection using multiple models and identification of VSN as a key regulator of mitochondrial homeostasis in hepatocellular carcinoma cells, and evaluation of growth differentiation factor 15 as an early biomarker of anthracycline-induced cardiotoxicity using an integrated PK/PD model. Trend 5 of 5, Cardiac Surgery Complications and Outcomes. This trend investigates complications, risk factors, and diverse outcomes associated with cardiac surgical interventions. This cluster covers 1,367 papers. Representative work includes diaphragm dysfunction after cardiac surgery, revisiting an under-recognized complication, prolonged mechanical ventilation and extubation failure in pediatric patients undergoing surgical correction for congenital heart disease, and preoperative diastolic dysfunction and perioperative risk factors as predictors of new onset atrial fibrillation in patients undergoing coronary artery bypass graft surgery, a retrospective cohort study. Up next, our first paper out of 5. Vaccination with mRNA-encoded nanoparticles drives early maturation of HIV bnAb precursors in humans by Jordan R. Willis and William R. She from Department of Immunology and Microbial Science, the Scripps Research Institute, La Jolla, California, 92037, USA. Let's break down its key contributions. Hello everyone. Today we're discussing a pivotal study in HIV vaccine research. Developing a vaccine that elicits broadly neutralizing antibodies, or bnAbs, is incredibly challenging but essential. This paper investigates whether a new mRNA-encoded nanoparticle strategy can effectively kickstart and mature these critical bnAb precursors in humans. This research involved 2 randomized, open-label phase 1 clinical trials, IVI-G002 in the US and IVI-G003 in Rwanda and South Africa. The studies evaluated novel mRNA-encoded nanoparticles, administered as both priming and boosting immunogens. The primary goals were to assess the safety of these vaccines and their ability to induce and mature broadly neutralizing antibody precursors, a critical step toward an effective HIV vaccine. The trials showed promising results. The mRNA vaccines were generally safe and well tolerated, though 18% of participants in the IVIG-002 trial did experience some skin reactions. Crucially, the priming immunogen successfully induced bnAb precursors with significant somatic hypermutation. Furthermore, heterologous boosting led to increased somatic hypermutation, improved antibody affinity, and enhanced neutralization activity, all vital steps in developing effective bnAbs. This study provides crucial clinical proof of concept, showing that mRNA-encoded nanoparticles can effectively drive the early maturation of HIV bnAb precursors. It's particularly significant that bnAb priming was demonstrated in Africa, where the HIV burden is most severe. While these are exciting early results, future research will focus on optimizing these boosting strategies and evaluating the long-term efficacy and safety of this promising vaccine approach. Up next, our second paper out of 5, small molecule targeting of the iron-responsive element in the AP mRNA 5-foot. UTR to control amyloid translation in Alzheimer's disease by Mateen A. Khan and Hassan S. Shaiba from Department of Life Science, College of Science and General Studies, Al Faisal University, Riyadh 11533, Saudi Arabia. Let's break down its key contributions. Alzheimer's disease, or AD, is a devastating neurodegenerative condition characterized by the buildup of amyloid-β, or A-beta, plaques in the brain. While A-beta has long been a primary therapeutic target, many drug trials have unfortunately failed. This paper explores a new strategy. Instead of targeting the A-beta protein itself, what if we could stop its production at an earlier stage by targeting the messenger RNA that codes for it? This review focuses on a fascinating biological mechanism, the iron-responsive element, or IRE, found in the 5' untranslated region of the AP messenger RNA. These IREs are unique 3D structures that regulate protein synthesis. The proposed design involves using small molecules to specifically bind to this AP IRE. By doing so, the goal is to disrupt the translation process, effectively reducing the amount of amyloid precursor protein and, consequently, amyloid-β that gets produced. The paper identifies the AP mRNA 5' UTR IRE as a critical and druggable target. It emphasizes that these IREs possess conserved 3D structures, making them amenable to specific small molecule binding. By targeting these structures, the proposed mechanism is to directly inhibit the translation of the AP protein, thereby reducing the generation of amyloid-β. This represents a novel and promising strategy in the fight against Alzheimer's disease. This research opens up an exciting new avenue for Alzheimer's treatment. By targeting the AP mRNA IRE, we could potentially develop drugs that prevent A-beta formation at its source, offering a fresh approach where many protein-targeting therapies have struggled. While this is a conceptual review, the structural insights provide a strong foundation for rational drug design. The next crucial step will be the development and rigorous testing of these small molecule inhibitors in preclinical and, eventually, clinical studies. Up next, our third paper out of 5, Artificial Intelligence-Guided Personalized Gut Microbiome Modulation for Persistent Secondary Gastrointestinal Symptoms in Oncology Patients. Clinical Efficacy and Biological Correlates from a Prospective Validation Study by Radu, Dumitru Dragomir, and Dorel Ionel Popoisi from Department of Oncology, Faculty of Medicine, Victor Babes, University of Medicine and Pharmacy, Eftimi Merga Square 2, 300041 Timișoara, Romania, RDD, DLS, AD, DIP. Let's break down its key contributions. Imagine completing cancer treatment, only to be left with persistent, debilitating digestive problems like bloating and diarrhea. This is a reality for many oncology patients, and current treatments often just manage symptoms without tackling the root cause. This study asked a crucial question. Can a personalized approach, guided by artificial intelligence, truly improve these long-lasting gastrointestinal issues by targeting the gut microbiome? And what biological changes accompany these improvements? To investigate this, researchers conducted a prospective, single-arm validation study. They enrolled 29 adult female oncology patients who had been experiencing persistent GI symptoms for at least 3 months. Each patient received individualized dietary, supplement, and lifestyle recommendations generated by an AI-guided platform. The study tracked changes in stool frequency, energy levels, and biological markers over a 3-month period. with each patient serving as their own control. The results were quite striking. After 3 months, patients saw a significant decrease in stool frequency, dropping from nearly 5 episodes per day to just over 2. Their self-reported energy levels also dramatically improved. These clinical benefits were supported by biological changes, including an enrichment of beneficial gut bacteria like butyrate producers and improvements in metabolic markers. Notably, increases in Bifidobacterium longum were independently linked to better stool frequency. This study suggests that AI-guided personalized microbiome modulation could be a game-changer for cancer survivors struggling with chronic GI issues, moving beyond symptomatic relief to address underlying biological causes. However, it's important to remember this was a single-arm study focused on female patients, so more diverse and controlled research is needed. The next step is to conduct larger randomized controlled trials to definitively confirm these exciting findings and integrate this precision approach into supportive oncology care. Up next, our 4th paper out of 5, unsupervised risk factor identification across cancer types and data modalities via explainable artificial intelligence by Maximilian Furl and Maximilian Merz from Scalable Data Analytics and Artificial Intelligence, SCADS.ai, Dresden/Leipzig, Universität Leipzig, Leipzig, Germany. Let's break down its key contributions. In clinical decision-making, accurately stratifying patient risk is crucial, yet current methods often fall short. They can be complex, difficult to interpret, and don't always translate into clear, actionable insights for patient care. This paper addresses this significant gap by introducing a novel, interpretable AI approach designed to overcome these limitations and provide more practical guidance. Our study developed an innovative AI method centered on a novel differentiable log-rank loss function. This technique allows neural networks to directly optimize for survival differences, enabling robust patient stratification. We rigorously tested this approach using diverse datasets, ranging from synthetic simulations to real-world clinical data from multiple myeloma and non-small cell lung cancer patients, showcasing its broad applicability. The results were compelling. Our method achieved highly significant patient stratification in multiple myeloma, with a p-value of 3.3 times 10 to the power of -27, and maintained strong performance during external validation. In simulations, it demonstrated excellent accuracy with an OROC of 0.94. Crucially, the model's decisions were interpretable, identifying clinically meaningful features that aligned well with established risk factors. This research offers a powerful, interpretable AI tool that can revolutionize patient stratification in oncology. By providing clear, actionable insights into patient prognosis, it promises to significantly enhance personalized treatment strategies. While highly promising, further research, including integrating more data types and conducting prospective clinical trials, will be essential to fully realize its potential in clinical practice. Up next, our 5th paper out of 5, Temperature as a Metabolic Signal Linking Neural and Endocrine Circuits to Energy Homeostasis by Shuiying Mo and Shengjie Fan from School of Pharmacy, Shanghai University of Traditional Chinese Medicine, Shanghai 201203, China. Let's break down its key contributions. Good morning, everyone. Today, we're diving into a fascinating review that explores how ambient temperature, a constant environmental factor, profoundly influences our body's energy balance. This isn't just about feeling hot or cold. It's about how temperature acts as a sophisticated metabolic signal, orchestrating complex neural and endocrine responses. Understanding these intricate pathways is vital, particularly as our modern, climate-controlled environments might be impacting our natural metabolic adaptability. Ultimately, this research aims to pave the way for innovative treatments for conditions like obesity and type 2 diabetes. This comprehensive review synthesizes a vast body of research, focusing on the intricate neural and endocrine circuits that connect temperature to our body's energy regulation. It delves into how peripheral thermal cues, detected by sensors in our skin and organs, are relayed and integrated by key brain regions like the preoptic area and arcuate nucleus. The review highlights how these signals then orchestrate critical physiological outputs, including feeding behavior, thermogenesis, and hormonal responses, drawing on insights from advanced circuit mapping and single-cell profiling techniques. The review reveals that temperature is far more than just a sensation. It's a powerful metabolic signal. It details how specialized peripheral sensors, like TRP channels, detect thermal changes, which are then relayed to central brain regions, such as the preoptic area and arcuate nucleus. These brain hubs act as sophisticated integrators, coordinating a wide array of physiological responses, from brown adipose tissue thermogenesis and blood vessel regulation to feeding behavior and hormonal outputs. This integrated understanding opens exciting avenues for developing novel therapies for metabolic disorders. So what's the big picture here? This review underscores that temperature is a fundamental, yet often overlooked, regulator of our energy balance, holding significant therapeutic promise. It also raises a critical point. Our constant exposure to thermoneutral environments might be contributing to metabolic inertia, suggesting that some thermal variability could be beneficial. Looking ahead, the challenge lies in translating these fascinating rodent-derived insights into effective human therapies. Future research needs to validate these mechanisms in humans to unlock the full potential of temperature-based interventions for conditions like obesity and type 2 diabetes. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.